Thank you very much. It's a great honor and pleasure to be here. I'm actually not quite sure whether I should be happy that my son will not become aggressive or whether I should be worried that I'm losing an argument against him. So, functional anatomy of episodic memory and its impairment in Alzheimer's disease. The studies that I'm going to tell you about have been conducted in the context of the German Center for Neurodegenerative Diseases, usually involving several centers. So mostly multi-center studies. And as you all know from the great Andrew Tulving, episodic memory refers to the ability to relive and replay past experiences. And so we want to understand how clinical Alzheimer's disease impacts on episodic memory. And what I will talk about is basically really our first data from DZNE, from the studies in the German Center for Neurodegenerative Diseases on this topic. I will also talk about the idea of maintaining episodic memory function in old age, so directly relating to the previous topic of maintenance. Then briefly talk about vascular reserve. This is something that we have been quite interested in, particularly in the context of exercise. Although I'll not talk about exercise, it turned out to be a topic that seems to be of broader relevance, so I'll talk about that. And then I will conclude with citizen science, which I think is a great approach to address some of the questions that were raised here regarding, for instance, lifestyle and cognition in old age. So when we think of episodic memories, we usually first think of processes. And of course, we all know the hippocampus, the surrounding entorhinal cortex, the perirhinal parahippocampal cortices, they are organized into circuits to which we can assign certain processes, such as pattern separation, pattern completion, and so on. But there's another organizing principle, and these are representations. And this is also, meanwhile, quite well established that the organization of parahippocampal, perirhinal, and entorhinal cortex follows also a representational theme in the sense that the posterior medial part of entorhinal cortex, together with the parahippocampal cortex, seems to be more interested in scene information, whereas the anterior-lateral entorhinal cortex and the perirhinal cortex more interested in object information. So when we look at the effects of Alzheimer's disease pathology on episodic memory, I think we have two dimensions. One are related to representations, and the other are related to processes. Now, usually people who have looked at processes were actually quite agnostic to representations. And in fact, representations were deliberately kept out of task design. So when tasks on pattern separation or pattern completion or associative binding and so on usually were very symbolic in order to avoid any representational structure. I think the way we have basically designed the studies in the DZNE is quite the opposite. So we focus both on processes and representations, and here particularly on objects and scenes. Now you also know, of course, that the two prevailing pathologies we have to think about are tau and amyloid, and we also know from the work of Heiko Braak, and this is postmortem work, that there is a lot of hidden, if you wish, pathology also in all cognitively normal older individuals. In the upper row, you see that a very large proportion of older adults with 60 have stage 2 Braak pathology in the medial temporal lobe, and also 30% of cognitively normal older individuals can have amyloid pathology in their brain. So it's clearly important to understand the impact of this pathology on episodic memory, both in healthy individuals, but also of course, as we progress, as cognitive deficits progress to mild cognitive impairment or Alzheimer's disease. So the study I'm going to talk about is the DEL-code study, where across Germany now over 1,000 individuals have been recruited, ranging from cognitively unimpaired to individuals with mild cognitive impairment to early AD. The numbers are not quite important yet because we haven't actually analyzed the full data set, although everyone has been now recruited into the baseline and also have several years of follow-up. So I can because there's a very strict data embargo and release policy, can only show you some results from sub-cohorts of this. Okay, so the way we look at episodic memory is at these functional networks is with task fMRI. So task fMRI is implemented across several sites, harmonized excessively. And we have two tasks. One basically contains complex images that have both scene and object information in them. So these are basically photographs of indoor-outdoor scenes. And the other task breaks them down into scenes and objects. So I'll first talk about the second task, the object and scene task, basically. These are the publications that kind of both led up to the work, but also entail the results that I'm going to show you about. And much of the work has actually been done by Emrah, who's sitting here. The prominent hypothesis that we wanted to test is that whether object and scene of information are differentially affected by amyloid and tau pathology. And the reason for this hypothesis is that the perirhinal anterolateral EC section is overlapping more strongly with the early stages of tau pathology. So we thought, there may be a privileged impact of tau on object processing. And for that, we designed a task, which is a mnemonic discrimination task using either objects or scenes. And you have to basically keep an object in memory, and after a while you see a similar object and you have to say, is it the same or not? And you have to do the same with the scene where the boundaries are changing. And now we compare this to Alzheimer biomarkers obtained through cerebrospinal fluid taps. And so the first data from Delcode, and this is already published, so you can read the details, actually show that the phospho-tau levels in the CSF correlate with the performance, so with the false alarm rate in the object task. And that's significantly stronger, the correlation, than the one for the scene task. We have also fMRI data, so there was an interest, of course, to look at whether the fMRI responses in the hippocampus are also affected by tau pathology. And here we see that there's actually a positive relationship in the sense that higher tau pathology is associated with higher activation. Now, this doesn't necessarily mean that tau leads to kind of a pathological activation. There can be many reasons why there is hippocampal hyperactivity. And it's not quite clear what the mechanism is, but one possibility is that the inhibitory input is disrupted from entorhinal cortex to the hippocampus. The other is that there is some impairment in one subfield which is compensated by another subfield. And there could be also intrinsic network hyperexcitability. So we don't know what the mechanism is, but this is an emerging picture that we have to really follow up. Now, Emrah did a very similar study, but now with tau PET in another cohort, in the Berkeley aging cohort. She quantified tau burden in an anterior temporal and a posterior medial network, here highlighted in colors. This is also published. And she found overall quite a similar picture in that the anterior temporal tau burden was related to object rather than disproportionate impairment in the object and the scene condition, using a very similar task. And not shown here, she found an opposite relationship with amyloid burden in the more posterior network. So there is a converging picture that tau pathology in preclinical Alzheimer's disease, and this is actually mostly also not including MCI individuals, but really cognitively normal individuals, initially affects object mnemonic discrimination rather than the scene representations. Now, looking at complex images, we think is a good way, because they entail both objects and scenes, to actually probe the activity of the entire network. And therefore, looking at photographs and how they are encoded and later recognized is for us kind of a nice screening test to now basically look at the activity of the entire network. And because we can probably look at the entire activity of the entire episodic memory network. We think this may be a good task also to look at maintenance of activity as you age. The task that we have implemented for that reason is a very simple one. You basically look at these photographs. Some of them are repeated over and over again. So you can look at repetition in the fMRI. And the instruction to the participant is to just make an indoor-outdoor judgment. It's very simple. And then an hour later, there's a recognition memory test. So the recognition memory accuracy, as you can see here from these numbers, as you go from healthy to subjective complainers to MCI to AD drops. There's also a relationship between recognition memory accuracy and tau burden. But what's more important for this talk now is basically what happens to the episodic memory network maintenance exactly in the same way maintenance is defined, very similar to how it was defined previously. So preservation of neural resources over the lifespan. So we thought, if this is the activity that typically the network, and we know that this is a network that young adults typically activate in this task, then we can take individual activity levels from older adults and compare each individual image to young adults, and then we can basically sort individuals in terms of their similarity to the young activity, to the young canonical network pattern. So this means we give basically every individual a single score. We call that just the fate score. And that single score tells you whether the episodic memory network activated is highly similar to young adults or dissimilar to young adults. So we basically, using that single score, describe maintenance, if you wish, in the sense that having a similar network means you have maintained something functionally. So there can't be a strong, lesion in the network, otherwise you would have activated a different network. So what I'm going to show you now is basically how this fate score across the participants in the DELCODE study relates to certain measures and to biomarkers. So here you see that the fate score on the y-axis is correlated with D prime, so there's a loose correlation, so that's nice. You can also see that it basically is associated with age. So the older you are, the more deviant your activity pattern is. But what's more interesting for us, of course, whether, how this is affected by tau and amyloid. And it turns out that basically it's the A-beta burden that is related to the fate score. So the more, the stronger the A-beta burden is, so this is A-beta 42 to 40, the lower or the less similar your brain is to that of a young person. And when we do a linear regression type analysis where we have the predictors of the fate score and with these variables, the one that really explain most of the variance is amyloid, followed by age and gender. Other cognitive abilities, such as word recall, don't explain so much variance, and also tau doesn't. So it looks like that the amyloid burden actually shifts the network activity pattern to one that's a bit that's more dissimilar to that of young persons. So this is now a very good starting point to actually look at what's underpinning this structurally, and you can now use maintenance as a starting point to look at things like compensation and so on. So this is an interesting starting point for a number of follow-up questions now. So in the next couple minutes, I want to talk about vascular reserve and the hippocampus. So of course, we all know that vascular risk is a very important factor that modifies how tau and amyloid burden impact on episodic memory. The reason why we were actually interested in this was that in old age, exercise often has no or even a negative effect on cognition. And we thought maybe we are overburdening the hippocampus vascularly, and we should really look at the vascular supply in the hippocampus in each individual. And turns out that that's possible. So with 7 Tesla, you can actually look at the vascular supply using time-of-flight angiography with a very high resolution required of 280 micrometers. And there are two there's one overarching principle here. So there are two supply sources. One is the blue one, it's the posterior cerebral artery, and the other is the red one, that's the anterior choroidal artery. So these are basically two different supply systems to the brain. But the important point here is that not everyone or not every hippocampus has an anterior choroidal artery supply. There is known inter-individual variability. This has been quantified histologically, and so we thought, we would like to see whether we can replicate the histologically known individual variability. And in fact, we can do this. Oh, sorry. So there should be actually another image. So this is the protocol for this is published in Neuroimage Clinical. So we can replicate the exact expected frequencies of individual variability in terms of the presence or absence of dual or single supply. Now the question, of course, is, and this paper will come out in the next couple of weeks. So Valentina Perosa asked the question, so if we take all the individuals who have vascular risk factors, and then, in this case, people with cerebral small vessel disease, and then look whether the presence or absence of of an anterior choroidal artery makes a difference. So that was basically the question. The outcome was it does make a difference. So you have a smaller anterior hippocampal volume with voxel-based morphometry, and you can see here the CVLT learning curve. So you learn. You are not as good when you have compared to those individuals who have a dual supply in the hippocampus. So that's something quite exciting, and we want to follow it up also in exercise studies. Okay, now I'm coming to the end. So some of, and I'm sorry, I should have mentioned in the initial slide, and a disclaimer of mine, I'm a co-founder of a company who has basically taken some of the tests that you have just seen into a mobile version, into a mobile app. And the reason why I want to mention it here is because, of course, there's a strong interest in the DZNE on mobile assessment, but also in citizen science. And there's mobile cognitive assessment with tests where you can actually link the results to known biomarker levels is something that I think is very important if you want to understand all the different factors, the multitude of factors that basically play or modify aging. And we have actually done a citizen science study in the DZNE together with the Ministry of Health, Education and Technology, where we wanted to look at the effects of flu in the winter months on cognition. We had basically, there, of course, see, the animation didn't really work. So within 4 months only, we could basically have roughly 1,800 individuals who completed 7,900 individual tests on mnemonic discrimination, pattern completion, and so on. And you see, for instance, here just the relationship to age and learning effects across trials and so on. With a very interesting, very nice age distribution, peaking exactly at the age that is actually relevant for us, 60 to 70 years. Now, the data are not fully analyzed yet, but I think the point here is that this actually shows that it is possible to do these types of studies and look at lifestyle-related risk factors with this type of approach. So I find that also very exciting because I think it's a direct translation from our research to involving citizens in answering some of these important questions. With this, I would like to end and highlight particularly two people, David Byrne, who is not here, and Anna Maas, who is here, who have really contributed to the studies I presented. Thank you very much.
Thank you, Emrah. Session is open for questions.
Thanks very much for a great talk. I was really interested in this data on the hippocampal vasculature. now you're focused on whether having an anterior carotid artery or not, but of course the circus of Willis is so variable anyway, and you have people that have, for example, a very prominent posterior communicative artery, so might actually, and I think they do, at least in the normal healthy brain, compensate perfectly well for any lack of that and that artery. And also, I think what is probably more important than just if you have this artery or not would be if there's like pre-stenosis or macroangiopathy that actually impairs the flow to this area. Because as I said, yeah, if it's a healthy brain, it's enough to have this one artery. Becomes much more severe if you have macroangiopathy and lack this. Do you have the amount of data to look into this in more detail?
Not yet. So this is actually quite difficult to do because it requires 7 Teslas. So it was quite a lot of work to get these individuals who are, of course, quite affected into the 7 Teslas. So I think what we need is somehow a way to translate this to a protocol that also works at 3T in order to be able to address all these questions.
Okay, thanks.
Maybe just adding to the vasculature group around Rö and Hassan in our department, they looked at people doing marathon. And they started and then sort of did a follow-up of the whole run, and then afterwards, I think 6 weeks, 3 months, and 6 months. And they looked at the vasculature, looking at the eye background. And it was interesting that against what I thought, marathon improved cognition, and that was highly correlated, 0.6, with a change in vasculature. So that might be something else beside the stable anatomy, that there is change over time in the things we do, which is, I found interesting.
Yeah.
Thanks for a great talk. You mentioned at some point during your talk, when we were focusing on the hippocampus processes perspective and the representations perspective, and then you focused on amyloid and tau. And I'm just wondering whether you already have started to look at representations, I don't know, using decoding things or others, and whether they also might be affected by pathology.
Yeah, so I mean, so far we've just looked at representations in terms of different, let's say, modalities or classes. But what would be really exciting is to actually look what's the quality of information that's actually represented in the activation. Yeah, I hope Anna I'm sure she will do that at some point, but she has so much on her plate.
So you can ask one more question with regard to the vascular changes that you showed. So I'm kind of wondering, particularly also about the hypothesis that exercise should even impair hippocampal volume because of changes to the perfusion maybe or vasculature, because the animal literature is quite heavy on studies showing that exercise leads to a release of VEGF, so vascular endothelial growth factor. So at least I would hope that if you have very severe problems in your vessels, that more exercise would actually lead to an increased vessel size, to more capillaries, so actually improves this problem. What do you think?
Well, I think a really consistent finding is that perfusion changes. In the study that we did, for instance, where we looked with gadolinium perfusion at 3T and then at volume changes in the hippocampus after exercise in old age, they are very highly correlated. So higher perfusion means higher volume. And but the problem is that the correlation, involves individuals who have smaller hippocampi after exercise, as well as individuals who have larger hippocampi after exercise. And the same is true with gadolinium perfusion. So across studies, I think this, the offset of this, the number of people who show negative effect varies a bit, but there is always people who show negative effects. Now, you could argue that's noise, but the reason why we were worried was that those who have the negative effects also tended to have poorer memory. It wasn't like maybe there's measurement noise and they improved anyway, and so there was actually quite a good relationship also between these physiological measures and memory changes. That was already a couple of years ago, and since then we said we have to understand, really we should look whether this is just noise, due to a small effect size, or is it really a true deterioration? And then we said, okay, the only reason that we can think of at that time as an individual driver of this was this vascularization pattern. So that's how it all came about. I'm not saying, just to be sure, I'm not saying that exercise leads to hippocampal atrophy. I would promote the opposite. But there are individuals who don't benefit and who do show smaller hippocampi after exercise interventions.
Okay, so thank you, Emrah.
Thank you.
We have to move on. And